Hi, everybody. This is Dr. Edward Park from Recharge Biomedical, and today we're going to discuss stem cell biology, and we have a special guest, Dr. Michael West. The two characteristics of stemness that people often cite are, number one, they're self-immortalizing, which means that they can reproduce millions and billions of times, and secondly, they divide asymmetrically. And what that means is, on the right, you see a picture of a cell dividing. It's a mother cell. And indeed, the term in Spanish is célula madre, which means mother cell, instead of stem cell in English, which implies more of a branching differentiation. So what happens is the mother divides into a perfect copy of herself and something a little more differentiated. So it's just like a master copy, like you would make a master copy of dollar bills or maybe vinyl records from your master copy. This is just two of the characteristics of stemness. I like to think of it as sort of like if you're on a desert island, like Lord of the Flies. If remember that book? You've got a group of cells, and somebody has to be the leader, hold the conch shell, so to speak. So what happens in any niche or population, self-sustaining island of cells, there's always a leader. If you look on the left, the zygote, which is basically the fertilized sperm and egg that your mom and dad made you, the zygote is immortal, and it can become everything. So all of us came from one cell, and quickly over days we became a ball of cells, and then a hollow ball of cells, and then a hollow ball of cells with this little thingy forming, which is comprised of three layers. And I know this is high school biology. It could maybe not be that interesting to people, but from the terms of differentiation, it is interesting because there are three layers. There's the inside layer, the endoderm, which makes things like lungs, pancreas, thyroid cells. Mesoderm, middle layer, which makes cardiac muscle, certain parts of the kidney, red blood cells, smooth muscle. And then the endoderm, the inside, or the ectoderm, which makes skin, nervous cells, and pigment cells. And then there's this privileged also crest here, the germ cells, which are eggs and sperm cells. The reason this is important is because we think of cellular differentiation as a loss of potentiality. So if you just use this pluripotent stem cell for an example, this thing can become red blood cells, platelets, white blood cells, B cells, and T cells. So this is a stem cell because it makes a copy of itself and then one of these downstream, more differentiated cells. So we referred back to this slide. What is aging? Well, I think aging is the accumulation of DNA errors, mainly in telomerase-active stem cells. But it also is in cells such as skin cells, gut cells, any cells that need to reproduce a lot. Perhaps they're not immortal per se, but the things that would die during chemo are the ones that are telomerase active. There's this thing called replicative senescence, which means that every time you copy DNA, it gets 50 to 100 base pairs shorter. And if you put skin cells into a dish and let them grow, they will only divide 60 times or so, the so-called Hayflick limit, before they acquire so many genetic errors that it's no longer viable. And this was sort of ignored by everybody at the time, as most paradigm shifts are. So it was only rediscovered or revisited by a Russian theoretical biologist who figured there must be something to lengthen them. Otherwise, how could we be sustained? If all the cells were immortal, then we would become these large blobs. So it's good that there are only certain stem cells, and the rest of them are doomed to die within 60 generations. So what is telomerase? Telomerase is this sort of engine that's always running to lengthen the ends of your chromosomes, these protective caps called telomeres. And it is how stem cells self-immortalize. And as a subset of that, we would consider cancer stem cells, which is the theory that most cancers or some cancers are derived from already immortal stem cells, those also self-immortalize. So if you look at it like sort of like Jimmy Olsen versus Clark Kent, well, if Jimmy Olsen decides to go crazy and destroy, you know, the city, that's not a big deal. But if Lex Luthor or Clark Kent does, that's a big deal, and that can become a clinical cancer because they're already super powerful and self-immortalizing. So in all of your organs, you have these niches where there's one cell responsible for reproducing herself, and the interesting question is, where do these stem cells come from? Are they promoted from within the company, meaning if she dies for whatever reason, are one of the drones made into the stem cell promoted, like by feeding them royal jelly? Or do they come from elsewhere, which could be the case in certain kinds of sort of traveling itinerant mesenchymal cells, which can repopulate organs? So that's an interesting question. We don't really understand how these cells interact with each other to determine who's going to be boss. One thing that's interesting is the cell cycle. Up until maybe a decade ago, people thought the DNA was kind of the be-all and the end-all. But now in the last several years, there's been a revolution of thinking, which is that of epigenetics and gene expression. So G2 is after all the DNA is copied, we also make sure that the software is correct. So if every cell is like a PC off the line, that would be the DNA. They're all the same. They all have the same library. But the epigenetics, meaning outside the genes, are all these modifications which unfold, which promote and which repress certain gene transcriptions. So it's really the software. It's the operating system on the PC. And if you look at the top 10 papers of any science year, several of them will be about epigenetics because it's so interesting, such a paradigm shift. We have these RNA molecules, which huge percentage of the genome was thought to be dark matter or junk. Turns out that that's not true, that these are very important antisense and certain binding proteins that are called small interfering RNA, which is probably another misnomer. There's things like methylation, phosphorylation, acetylation, which do little flags here on the DNA. The chromosomes fold and unfold. Here's an example of that. Here's a gene in red, which when it's the DNA is wrapped in these little spools, and then the spools are bundled together. So here, the gene in red can't be transcribed, it can't be accessed. But with certain epigenetic factors, the spools unwind, and this gene would be available for transcription. So epigenetics just is a catchword for how the cells express their genes and how they express their role in life, their differentiation. Big revolution is IPS, which is induced pluripotent cells. What they did several years ago were some scientists expressed some genes by introducing a virus, and they took a regular skin cell and they reverted it back to a pluripotent cell, or one that could become pretty much every line of cell, endoderm, mesoderm, and ectoderm. So turning now to Dr. West, who's shown here. He graduated from RPI in psychology and then got his master's in biology at Andrews University, and finally his PhD in cell biology from Baylor. And a year later, he actually founded what is a $500 million company now named Geron, of course derived from the term gerontology or age. And his mission at that time was to understand, by virtue of telomerase and the things that we did know at that time, which I think it was just maybe several years after telomerase was discovered by then to be Nobel Prize-winning Blackburn, Greider, and Shostak. He decided to get some people together and found a company which would really dominate intellectual property of stem cells as well as telomerase-related patents. During that time, he was instrumental in working with James Thompson at the University of Wisconsin, and they were able to isolate human embryonic stem cell lines, which, due to social and ethical concerns during the Bush tenure, were sort of limited. So not really much clinical research came of that because they felt that using sacrificed embryos as a source of these pluripotent cells was questionable. although Geron finally last year did have a human experimentation of spinal cord injury in one of their lines that Dr. West was instrumental in forming. From 98 till 2007, he was at the helm of ACTC, Advanced Cell Technology Corporation. And since leaving there, he has gone on to form Biotime, which is a multi-branch sort of regenerative medicine pioneer. One of their main flagship products is Hextend, which the military uses as a blood expander. When you have volume loss, you can put this Hetastarch, which is basically, Gatorade for the blood with some osmotic components. Makes sure that you can get blood necessary to all organs as you're exsanguinating. It's one that's used in surgery and throughout battlefield conditions. He also is the principal in Biotime Asia, and they're experimenting in stem cell therapy and also in treatment of certain tumors. Finally, Embryome Sciences. They have a very interesting portfolio of progenitor cells, which we'll discuss. And progenitors are not totipotent, meaning they're not all potent cells, but they're differentiated a little bit down a line. So if you need something for the heart or, say, for the liver, there's 100 different off-the-shelf things you can go and buy on the website, which will be used for research and possibly therapy. They also have different products for growing and sustaining stem cells so that they don't differentiate. They've got this fascinating, almost bioluminescent technology where they can show when a gene gets turned on, it'll actually glow. And they're also looking into this black magic of growth and differentiation factors, which is really what we're hoping to understand more. The cells, when they're forming a creature, or they're repairing or just functioning, they know how to signal each other. And so hopefully, stepwise, Embryome Sciences and other researchers are learning to turn these switches one step forward, one step back, all those other epigenetic signals that we discussed. Dr. West, can you hear me?
Can you hear me?
Perfect. Yes, I can.
Good.
Okay, great. So that was kind of just a quick overview of what stem cells are. Welcome to the call. Thank you for being on.
Well, it's my pleasure. Interesting discussion so far.
Great. So obviously there's so much we could talk to you about the whole history of Geron and the Bush era, and but I think that's, although interesting, not as interesting as some of the basic science things that you're into. A lot of people I know, patients of mine, have gone to Swiss Clinic or what have you to get black sheep stem cell transfusions. There have been offshore India, say maybe Venezuela, China treatments, which are questionable.
I think many of the listeners here on your conference call are scientists, and I think it's important for us in the scientific community to speak out a bit about this issue. Every such product that I have personally looked at, without naming any particular company, is nonsense. It's very clear. All except for old world primates have glycosylated changes on the extracellular surface of cells that causes hyperacute rejection.
Do you have an official position, either as a scientist or as a CEO of Biotime, regarding telomerase activation and its safety?
Well, it's of great interest to me as a scientist. I think that there's a significant amount of evidence now that telomere damage or telomere shortening with progressive cell lifespan is playing a fundamental role in aging. I would point to the similarity of Werner syndrome and Hutchinson-Gilford syndrome, progeria, with human aging, and the now substantial amount of data that these inherited diseases are linked to telomere biology, and that telomerase can rescue that phenotype.
Say you have retinal pigment cells that are burnt out. To take a good copy differentiate it in a purposeful, stepwise manner, and then transfer the nucleus back to the existing, or just maybe killing the existing retinal cell and replacing it with a brand new self-derived stem cell. That kind of personalized stem cell. Are we going to see that soon? Because presumably we have a pretty good copy of our original library in the spermatocytes and oogonia and things like that.
I came to the conclusion that clonally isolating or expanding purified embryonic progenitor cell types and using them to repair case by case damaged skin or bone or cartilage or heart muscle, the midbrain in the case of Parkinson's, the RPE cells of the retina for macular degeneration, is sort of like repairing an old antique car, keeping it around by replacing defective components. I saw that we could unlock the natural telomerase positivity and the regenerative pathways in early development to allow this field to make some significant advances in the biology of aging.
I mean, I think the induced pluripotency is interesting from a scientific standpoint in that it kind of, in a reductionist way, aims to the mechanisms that help maintain, potency and lack of differentiation. But, I read a paper recently that suggested that when you do make these differentiate cells look more like pluripotent cells, that you have a lot of things that are not reverted.
What's remarkable is that 4 transcription factors can do so much. The reprogrammed iPS cells with just 4 transcription factors are indistinguishable, in our experience, from normal embryonic stem cells.
I think that the epigenetic signatures, are those reset? Are those rebooted as well? Because to me, it's more like a Liza Minnelli facelift. There's a lot of little other switches that determine, the function. How could you possibly reset those unless it was like a total?
I don't know what the Liza Minnelli facelift means, but my point was that in terms of gene expression, it's just amazing that you can get as close as we already are. Now, in regard to imprinting, there's still a lot of question marks here. There's a lot of contradictory data out there.
Hi guys, can you hear me okay?
Yeah, perfect.
Fine.
Hey, Mike, first of all, you probably don't remember me, but I did meet you at Manhattan Beach Conference. Greta introduced us. It's a great pleasure to listen to you. So thank you, and thank you for what you're doing. And I so look forward to having the parts bag. Also, I should shout out to both the Andrews, the senior and the junior, and thank them for everything they've done for me, and Ed, you too. Mike, I read your chapter in Greg Fay's book, and one of the things that I thought for me was a revelation was this concept of sense-static activation. Could you comment on that and its role in the aging process?
I appreciate the question. Years ago, when I was a graduate student working with Jim Smith down at Baylor Medical School, I was of the opinion that when cells aged, they must be exerting their effects by secreted proteins, because you see this degeneration, for instance, in the skin, a melting of the collagen in the skin, elastolysis, the destruction of the elastic network that's put in place early in development. And I thought, well, this has to be through metalloproteinases or some other similar change. And so I started culturing young and old cells, senescent cells, side by side and looking at the secretome, the radiolabeled secreted proteins. Young cells exit the cell cycle and then dramatically change the proteins they secrete. And when they're activated, like in a wound, they again change this secreted protein profile. Senescent cells were stuck in a non-proliferating state, but it was the activated state, like a young cell that's actively proliferating. So it's like a car with its accelerator stuck and its brake stuck down at the same time, a very abnormal state. And what a lot of people have never picked up on is that that is a fundamentally important explanation for much age-related degenerative pathology. We hear a lot about inflammation. Many diseases of aging they appear to be inflammatory. It's not that there are immune cells in many age-related damaged tissues. It's just that you see inflammatory-like processes. And what's going on is senescent cells that have telomeres have their accelerators stuck as well as the brake. And they're secreting high levels of metalloproteinases like collagenase that destroys the collagen in your skin, making it paper thin, and of course the elastic components as well. And it's these, I just dubbed this synstatic activation. The same thing occurs in cells with any kind of serious genotoxic event like double-strand breaks.
Thanks, Mike. And I just would point out for those who are interested in finding out how that might interact with the mitochondrial theory of aging, Rhonda Pino's papers recently, for me anyway, were quite elucidating in that they really tied the control and the effects of mitochondria to telomere length in terms of mitochondrial biogenesis, mitochondrial function, and also, interesting enough, mitochondrial mutation rate. So when I read this in the book, Mike, I said, you know what, this this needs to become, someone needs to get this out there, and I'm so glad that you did.
It needs to be discussed more because I think a lot of people see these inflammatory-like changes, Alzheimer's, the arthritis of the brain, and many of these age-related diseases are seen to be inflammatory-like, and no one's quite recognized that senescent cells have an IL-1-like pattern of gene expression. It's a self-induced inflammation, so to speak.
Do you think, Mike, anti-inflammatories of the common type, both nutraceutical and prescription, would benefit that type of?
Yeah, well, I think so. At least some. Remember, there's different inflammatory pathways and different agents that modify inflammation.
So, Dr. West, was there anything from the presentation that was not correct or maybe misleading, do you think?
No, I think we covered several of the important points. I would point out that the largest ethical problem we face in reality is the aging baby boom population. Our country has made a commitment under Medicare for over $70 trillion, that's with a T, $70 trillion to this generation. I noticed on 60 Minutes here recently, an episode, there was a discussion of this, and the experts were saying, look, we just need to set limits. We need to say that at a certain age, we'll no longer take care of people because we can't afford it. It'll bust the bank. That would be the first time we as a country ever made a conscious decision to discriminate against a group of people based on a biological characteristic, their age. And the solutions need to be technological, not the stroke of a pen in the general accounting office. But I'll get off my soapbox.
Yeah, that's a great point. And, the government can collect more taxes and never have to pay Social Security if they keep us healthy. So maybe some bean counter will figure that out.
Yeah.
I have Peter Douglas is on the line. He would like to ask you a question. Peter, can you hear us?
Yes, I can. Can you hear me?
Yes.
Yes, indeed. Go ahead.
Okay, excellent. Well, let me introduce myself first of all. I am the managing trustee of the Douglas Foundation, and we're a private family foundation. We've been around for quite a while, and over the past few years, we've begun to focus more and more in the areas of medicine. I'm looking for some expertise as to where those things actually stand and what the promising treatments are that might use our support. From my point of view, it's a question of how can you extend life and maintain its quality at the same time. And there seems to be a lot of promise in stem cells, and I just have no clue as to how to sort through the real promise from the backstreet, promises.
I think if you tune into our weekly shows, we're having, as Mike mentioned, arthritis is a huge problem. We're doing a show on arthritis from a TA65 user. Bone health, we're doing one on macular degeneration. And so there are a variety of topics related to aging and regenerative medicine. And in my biased opinion, I think that TA65, because it's a telomerase activator, does work on stem cells. And so it's stem cell regeneration, not as technically sophisticated as what Dr. West is doing, but trying to do it the natural way through your own mechanisms. So if you tune in weekly and listen and you think that there's some validity to what the people are saying, you could potentially set up a fund to do randomized controlled trials, and that would solve the problem for everyone. There would be real science, and the cost to null patent of extracting this, which is exceedingly high, will be deferred, and then you can get some kind of contribution to real disease states. I think that would be a good bang for your buck if you want to help us get the good word out.
Yeah, this is Noel Patton.
Okay, Noel Patton. I'm sorry, no, this is Noel Patton, the founder of TA Sciences.
Well, a couple comments. TA65 is we have the license for it from the Geron Corporation that Mike West started, and if I'm not mistaken, the basic premise of Geron at that time when it was founded was to, quote, cure aging. And we have so far the only product that's been commercialized out of all that Geron technology. We're actually selling a product that people can take that activates telomerase. And one of the complaints that we heard many times and came up here in this conversation was that we don't have the double-blind, placebo-controlled trials that we'd like to have. We have one published paper that just came out in September that's showing that human beings that took TA65, their shortest telomeres got longer and their immune system got stronger. We're right now in the planning stage for a 1,000-person clinical trial, 500 placebo and 500 taking TA65. These are going to be older people, and we're going to be looking for extension of life and a reduction of hospital stays. So the very, very important thing of what's been talked about here, we do have a problem. Willing to donate the TA65 for this trial, but we're a startup company that hasn't made any money yet, and we do need funding in order to pull off this trial, which would be earth-shaking because if it goes like we expected it will, we will show that taking a telomerase activator, which is in this case TA65, actually extends human life or improves the quality of human life by limiting the number of hospitalizations for old people. The question I'd like to ask Dr. West about is that Dr. Pino's studies there at Harvard, where they showed, at least according to what he's written, and these were published in Nature, which is a pretty serious journal, that the activation of telomerase was able to actually reverse aging. Wouldn't any telomerase activator do the same thing?
Yeah, I mean, I think generally speaking, the Pino paper was almost obvious from many people's opinion. It was a demonstration of something that, years ago it was demonstrated by our group at Geron, that the collaborators, that the telomerase could completely abolish the aging cells in vitro. And so we say it immortalizes cells, and it does not malignantly transform the cells. I think the best evidence is that the telomere is fundamental in many aspects of aging, and that reactivating telomerase can abolish many of those aspects of aging. Some things in aging are wear and tear. You break a tooth, telomerase isn't going to regrow a tooth. But combined with regenerative medicine technologies where we can regrow cells and tissues like teeth or other cell types, and telomerase activation, I think there's, again, a great opportunity in our lifetime to make inroads for this problem of aging we're facing acutely in the baby boom phenomenon.
What if what if we find a telomerase activator that is much more powerful than TA65, and you take it and it reverses cell aging very rapidly.
So the reactivation of telomerase could run the risk of causing malignancy. And Noel, as you know, I've been outspoken about those risks. But if the goal is to increase human life expectancy and health as long as is possible, we're going to have to balance that narrowing tightrope walk. And so the image I have is ultimately we're all walking this high wire. And the risks of eliminating the manifestations of aging is you're increasing the probabilities of cancer. And that's a balance we're going to have to very skillfully walk. It's in part why at Biotime we're targeting some of our technologies.
I do want to point out something, Mike, that you may or may not have heard. We now have well more than 1,000 people taking TA65, and we haven't had a single report of anybody who wasn't diagnosed with cancer being diagnosed with cancer, and those people who had cancer before they started taking TA65, we haven't had any reports of increased cancer load. And quite the contrary, in some cases we have seen remission and a reduction of cancer load. So I don't want to imply that we're preventing or curing cancer, but you'd think with 1,000 people at average age of 64 that the fact that people are not getting more cancer from taking TA65 is a pretty good indication that TA65 doesn't increase cancer load.
Yeah, no, as you know, we've debated this in some public formats, and my position has always been that for older people who are at risk of mortality or serious disease as a result of aging, TA65 has demonstrably been consumed for centuries in China. It's a natural product. You know, the risks are probably small. Your compound is highly purified, however, and that is not what was put in the teas of people for many years. But for young people that are otherwise healthy, being exposed to telomerase activator for a long period of time, my point of view, and it's just worth what it is, my opinion, is it may unnecessarily expose a healthy person to the risk of malignancy, in my opinion. And you could be quite right. We don't know all the biology. There's a basis to believe that reactivating telomerase can help actually prevent cancer. But my point of view is the reason it's in our body is it was selected ultimately as an anti-tumor mechanism to allow us to live as long as we live. So both are true at the same time. It sounds contradictory on that.
So the verdict's not in. We have had this debate, but my point on it is, or my view is that we know now from live humans plus other tests in the lab that telomerase activation, TA65 in particular, strengthens and rebuilds a weakened immune system that's been weakened by age and where the immune system cells have suffered telomere loss to get to a short critical stage. And it would seem to me, just as a layman, that having a strong immune system is the reason that, well, you just said all of us have cancer cells in our body. Hopefully most of us on this conversation are not suffering from an actual disease, and that's because our immune system searches out and kills those defective cells. So, the fact that we're strengthening the immune system is a pretty good thing, I believe, and that's where we actually have statistically significant evidence. That's where we send the blood to UCLA's immunology lab and measure telomere length. Measure the immune system in lymphocytes and granulocytes, and we're seeing a reduction in the senescent immune cells.
Okay, guys. I know you've had this Clash of the Titans debate before, so I think both of you scored your points, and we'll wait for the judges to end the round. Any other comments, Noel, that you want to make?
No, but thank you for doing these weekly things. I think it's I take my hat off to you.
Oh, it's fabulous. Thank you for actually listening in. I appreciate your time. It's very valuable. But I think you'll be excited to hear that in the next 4 weeks, I already have guests lined up for arthritis, macular degeneration, bone health, spinal health. So there'll be a lot of more interesting, real human voices and people that have TA65 in their systems and are pleased to talk about it. So I think you'll enjoy those too.
Well, thank you for your help in furthering people's awareness of telomerase activators.
Well, I mean, thank you, because I think, I've said this before, that there's really few people that could have gotten this to market except you, and I have you to thank. And I think a lot of my patients now, whether they know it or not, they think it's expensive, and I tell them it's expensive to extract, but it's really changing their lives, and I have no compunction advocating for it. I didn't want the stem cell webinar to be about that, but I do think and I think Bill Andrews agrees, that's where we're really doing the activations on our existing stem cells. So it's exciting. So anyway, I'm going to go ahead and sign off here. Bye, Noel.
Bye. Thank you so much. Bye.
Oh, my pleasure.
Thank you.
Dr. West, thank you so much for sharing your insights with us and your time. I know you got to go at 1, so I apologize for keeping you late.
No problem.
All right,
guys.
we've been talking with Dr. Michael West, CEO of BioTime and founder of Geron Corporation. My name is Dr. Ed Park from Recharge Biomedical, and please tune in to future webinars. We'll be discussing a range of other interesting topics. Thanks, everybody.